Stéphane Anger. I'm the Associate Dean of Research at the Faculty of Pharmacy, and I'm also the co-director of the PRiME initiative. And it's my pleasure today to begin this second half of the symposium with two features of biotechs in the Canadian ecosystem, two successful biotechs that have recently gone through the IPO process. And the first one we're going to hear is from Chinook Therapeutics. And we have the pleasure to have Tom Froelich, who is the Chief Business Officer at Chinook Therapeutics, as well as an entrepreneur in residence at Versant. So prior to Chinook, Tom was the VP of Business Development at Arbutus Biopharma, where he played a key role in multiple transactions, including the merger of Tecmira and Encore and the spin-out of Genevant. Previously, Tom worked internationally at J&J and Merck in various roles, leading commercial strategy across all stages of product development, including the global launches of Telaprevir and Stelara. Tom has a BSc in biochemistry from the University of Victoria and an MBA from the University of Oxford. So thank you very much, Dr. Froelich, for joining us, and please go ahead with your presentation.
Great, thanks so much, Stéphane. And I like the promotion to doctor. I'm not quite there yet, but maybe someday.
Okay, I'm sorry about this.
No problem. So thanks so much for the very kind introduction, Stéphane, and then thank you as well to the organizers, Christine and Shana, for inviting Chinook to present and attend. It's a really exciting time in the biotech ecosystem in Canada. There's so much going on, and it feels like a great time for momentum to build some of these companies. So I'm sharing my screen now. I'm just wanted to check to make sure that everyone can see.
It should be okay, Tom.
Okay, excellent. So thank you so much. So Chinook Therapeutics, as Stéphane mentioned, we recently became public, actually, of Monday of last week. So I'm just getting used to this. Our lawyers tell us we have to insert a slide like this now that talks about forward-looking statements, but we don't need to spend too much time on that. But Chinook Therapeutics was a spin-out of Versant Ventures, which many of you will be familiar with. Versant is an early stage life science venture capital investor, focused now almost exclusively on therapeutics in the biotech space. And Chinook was formed really with the idea that the time is now for drug discovery in kidney disease. Many of you will know that kidney disease is a huge burden and a massive problem for patients, but also for society. Right now, the current therapy for most progressive kidney disease is actually quite limited. It's limited to only using things like blood pressure lowering medications or glycemic control in the case where there is diabetes. But in most cases, it's almost palliative care, almost progressive or watchful waiting while disease progresses to the stage of dialysis or to transplant. And those are really terrible places for patients to end up. When we talk about dialysis with patients with kidney disease, one of our key scientific advisors, Matthias Kretzler out of University of Michigan, he likes to quote a stat where if a patient ends up on dialysis, their prognosis is actually worse than if you're diagnosed with metastatic cancer. Survival rates typically are around 50% at 5 years for somebody who starts dialysis, and those get even worse if the patient has diabetes. Unfortunately, despite that fact, there's very few drugs that actually prevent the progression of disease, as I mentioned. And the cost associated with treating end-stage renal disease is huge. It's over $120 billion a year in the US, just managing dialysis and transplant patients. So it's a horrible problem. Historically, the pharmaceutical industry has not been able to address kidney disease very well. It's been a really tough place to go after. And we looked at why the reasons were, and it seems historically that kidney disease was really hard to tease out what was happening. the kidney is a very complicated organ, over 30 different cell types, and it was really hard to understand what cell type was driving disease, and then what was what was dysregulated in that cell type that was causing disease. In addition to that, a lot of the diagnosis made in kidney disease was really made more by pathology or histology or symptoms rather than what is that underlying driver. So you ended up with a lot of heterogeneous groups of patients in trials, which always is complicated for drug discovery. But we think that's all changing. Over the last 5 or 10 years, a lot of academic groups have started to use biobanks to tease out exactly what is the underlying driver of disease, and starting to segment patients into cohorts where you can identify what is their underlying molecular driver of disease, either a genetic dysfunction or a key biochemical pathway that's dysregulated. So as drug hunters, we love that because that really increases our chances of success with a particular target. In addition to that, there's new tools, things like single-cell RNA sequencing that really let you tease out what is occurring within a cell, and then stratify patients. So when you do go into clinical trials, you can ensure that you're enriching them to get higher likelihood of benefit for those patients. So we love that. It's a big market, new targets to go after. And then the third factor that really gets us excited here is that the regulatory landscape is changing. Historically, as I mentioned, it's been a tough place to do drug discovery. One of the big elements there is when you're going after heterogeneous patient populations, it's really hard to understand if your drug is working or not. And regulators have historically always demanded that in kidney disease, outcome trials were done. So show that you're preventing time to dialysis or show that you're preventing time to transplant. And that's caused you to have to do trials with 5,000 patients and measure them for years and years. But now if you go to the regulators and go with a specific indication that's well characterized, so something like IgA nephropathy, where you can show what the final common pathway is for disease, they're showing a more of a willingness to accept registration based off of surrogate biomarkers. So things like reductions in urinary protein, or things like surrogates like eGFR, which is a measure of kidney function. So instead of having to do a huge 5,000 patient trial, you can get away with showing benefit in something like 300 patients over a much shorter time frame. So it's a very exciting time for drug discovery in kidney disease. So Chinook was set up to address that, really driven by deep insights into disease biology, focused on indications where there are rapid opportunities for clinical development and registration off of a surrogate biomarker. The team, as I mentioned, is based in Vancouver, a research group that came out of Inception Sciences, which was the drug discovery engine of Versant Ventures. And we wrapped that very talented group of drug discoverers with some top academics who use tools like single-cell RNA sequencing to tease out underlying biology, but also novel systems like kidney organoids that can be used to study human tissue in a Petri dish. Out of that, we have this middle column, which is our emerging pipeline. That's drugs that have come out of our internal research capabilities. And we have CHEK 336, which we anticipate having an IND in 2021. Then we have a clinical pipeline that actually was mainly acquired. Very early on, we became interested in several disease indications where you could apply this precision medicine approach to the kidney. And out of that, we became interested in several targets. The lead target that we became interested in was endothelin, which actually atrasentan is an endothelin inhibitor. And through our early research and using single-cell RNA sequencing, we understood that endothelin was highly implicated in disease progression in a number of kidney diseases. And we found some novel insights into the mechanism around inflammation, fibrosis, as well as the commonly known hemodynamic effects. So we got really interested in that pathway, but found that instead of inventing something ourselves, that there actually was a molecule sitting on the shelf at AbbVie. They had tried it in diabetic kidney disease, and for reasons that I could go into maybe during the Q&A, had decided to discontinue that program. So we managed to in-license it, and we're moving forward with a phase 3 trial for IgA nephropathy shortly. We also, through a merger, acquired Bion 1301, which is an anti-APRIL antibody that we're moving into a phase 1b trial currently. So this is what our pathway or our pipeline looks like at this point. As I mentioned, it's a very robust pipeline across a number of stages of disease. Atrasentan is, as I mentioned, we're initiating a phase 3 program in IgA nephropathy, and that's anticipated to get started early next year. But we believe that this mechanism can actually apply to a number of different proteinuric kidney diseases. So we're also starting a basket trial in a number of conditions that is anticipated to start shortly after the phase 3 trial next year. As I mentioned, this Bion 1301 is another agent that goes after the root cause of IgA nephropathy. And we're initiating a phase 1b trial that's actually ongoing. And we anticipate having patient data for that in the first half of next year. And then we're going after CHEK336, which is a primary hyperoxaluria, which is an ultra-rare orphan kidney disease. And we've nominated that drug as a development candidate and have plan to have a phase 1 initiated by the end of 2021. In addition to that, we have a very healthy research program where we are looking at a number of severe chronic kidney diseases, including quite a large effort around autosomal dominant polycystic kidney disease. And we've set up a number of innovative and translational assays and platforms, as well as a number of traditional ones as well. And we're looking at a number of targets for target validation and have one program in lead optimization. We're also looking at a number of other programs that we haven't disclosed yet in other severe rare kidney diseases. So I just wanted to mention a little bit about this idea of precision medicine and bring in an example of why we're going after IgA nephropathy. IgA nephropathy is a rare kidney disease, but it is the most common primary glomerular disease, affecting about 140,000 patients in the US. Patients are typically diagnosed in their 20s or 30s. And about half of them actually progress to end-stage renal disease in about 20 to 25 years. We think this is a really good program to go after, or indication to go after from this precision medicine point of view. Because if you look at the etiology of the disease, which has been teased out in the recent decades, it's caused by this aberrantly glycosylated IgA. IgA is the immunoglobulin that's actually present in mucosal tissues, so in the lungs and in the gut. And it seems like there's one aberrantly glycosylated form, which actually forms an antigen. And when you get this production of autoantibodies that binds to this aberrantly glycosylated IgA, you get an immune complex, which then deposits on the kidney. So we're going after this in two different ways. One is with atrasentan, which does go after the HIT4 here on this diagram, which is involved in inflammation and fibrosis and some destruction of the basement membrane here, which causes proteinuria. But we're also going after the root cause here with 1301, which is an anti-APRIL antibody, which does eliminate this glycosylated, deglycosylated IgA, which takes out the initial hit. So we are going after this in two different ways. We recently, I wanted to talk a little bit about in the few minutes I have left, some of the financial strength of Chinook. We recently did go public, as was mentioned in the intro by Stefan, through a merger with a publicly listed biotech company. It's been a very fast trajectory for us. We raised our initial Series A in 2019, backed by Versant Ventures as well as Appletree and Samsara. We announced the merger in June, and then completed a private placement, and then through the reverse merger, did manage to increase our balance sheet as well. So now we have over $275 million in operating capital to fund the aggressive pipeline that we have outlined. And we believe that that will take us into 2023, when we aim to have our readout of our phase 3 program. So just in summary, this has been a very rapid trajectory. We think there's a huge opportunity for taking that precision medicine approach, which was so successfully developed in oncology, and apply it here in kidney disease, where there's a massive unmet need. We founded the company in 2019 with this very entrepreneurial group out of Vancouver, and have grown now to, but we'll probably end the year with about 90 employees. But 2020 was really an area of growth for us. We did in-license AbbVie, as well as lay the groundwork for initiating this phase 3 and phase 2 trial with atrasentan next year. We're initiating the IND enabling studies for 336. And as I mentioned on the last slide, we secured the financing that allows us to execute on these ambitious plans. And then next year is really looking at execution. We need to get these programs up and running and enrolled, and start reading out on the data to get these medicines to patients. And then over the long term, we really want to be the leader in kidney disease. We think this is a huge opportunity to take this precision medicine approach into this disease area and really make a difference in the lives of patients. And the way that we plan to do that is continue to focus on these R&D principles of precision medicine, the way that we think about it, which is a key focus on specific kidney diseases where we can tease out that genetic driver of disease or that key biochemical pathway that's dysregulated. Focus on novel targets and novel molecules that can address those, and then go after populations with novel approaches where we can apply surrogate endpoints to make sure that we are bringing benefit to patients with kidney disease. So maybe I'll pause there. I think that I was supposed to go for about 20 minutes, which I think is about now, and leave some time for questions.
All right, thank you, Tom. So if anybody has question, please feel free to use the question and answer feature of Zoom. If in the meantime, while people are typing, I have one. So you mentioned that you have a number of innovative research programs at Chinook. I was wondering, and you also mentioned a number of innovative platforms that are being built. I wonder if you can comment a little bit on what those are, what strategies are you taking in order to research new targets, perhaps?
Yeah, it's a great question, Stefan. So our main thinking is that we want to focus on diseases where we can understand what the underlying driver of disease is, and where we have an opportunity not to have to run a huge clinical trial. So when we first set up the company, we did a filtering exercise where we looked at 150 different non-oncological kidney diseases and filtered through looking at which ones are severe enough that are worthwhile the effort, have enough of an unmet need without a good standard of care, and then looked at where we know we can find either a genetic or biochemical dysregulation, and then where we can develop with a surrogate endpoint to make sure we're not doing studies for years and years. Then we assembled our biology team to look at those things. And there are some traditional models that you can use, but we really have leaned very heavily on single cell RNA sequencing. We think specifically in the kidney, where there are so many different cell types, that this does provide a lens into disease that you don't get in any other way. And it's really allowed us to look at certain diseases where single cell sequencing has never been applied. For example, in polycystic kidney disease, it's very difficult to get human kidney tissue because biopsy is actually contraindicated. So we did work with our group of academics to find some tissue, and then work with Ben Humphreys at Washington University in St. Louis, who then ran the single cell RNA sequencing with us. And that's really allowed us to validate a number of targets that we were thinking about, and also give us new insights about why some drugs were working and some weren't, and did give us that novel angle. The other platform that we're putting together is the use of kidney organoids. And as you're probably familiar with organoids, you can take a single iPSC from a patient and then use those with a differentiation protocol to grow up a full nephron. And in drug discovery, as you're well aware, there's lots of assays. They all have their strengths and weaknesses. Getting that one more piece of human validation is just extremely useful. So we've been using organoids to in our drug screening programs. In PKD particularly, we've managed to get a cell line with polycystin 1 knocked out, and those organoids actually spontaneously form cysts, and it's been very interesting to work on, both from a target validation point of view, but also testing different mechanisms.
Thank you. Yeah, a good example of two enabling technologies that we've seen over the last 5 years or so taken up, in the pharma industry. So we have a question from the audience. I'm assuming there's multiple reasons, but can you comment on why atrasentan was not developed internally by AbbVie?
Yeah, it's a great question, and probably the most frequent one we get from investigators and investors in the company as well. So AbbVie did start this 5,000 patient trial with atrasentan in diabetic kidney disease. And they fully recruited it, spent, I think, 2 or 3 years actually recruiting all 5,000 patients. And then what happened at AbbVie, and I'm speculating slightly because I don't have all of the internal information, but what we believe happened was they had a full management turnover with their CSO and their CMO. And they ended up actually focusing on autoimmune disease and oncology, and decided to actually withdraw all early stage research from nephrology. And during that time, they kept the SONAR study running, where they were looking at diabetic kidney disease, but they were anticipating to have about 425 events in the trial. And when they looked into the first data cut with the data monitoring committee, they actually only had about 80 events. So a very small number. And they looked at the run rate and the trajectory, and they thought they could run this trial for years and years and never have enough events to prove that atrasentan differentiated from placebo in any way. So they ended up making the very difficult decision, actually much to the chagrin of the 800 investigators that were involved in that trial, to discontinue the study. The good thing they did, because the investigators were so vocal, was they did shut the trial down per protocol. And what ended up happening was, between the time they announced the shutdown and when they read out the data, they had actually 100 more events accrue. So they ended up with 180 events, which wasn't quite the 425 they were expecting. But in that time, when they analyzed the data, atrasentan actually had a bigger treatment effect than was expected. And the data came out positive. So I think, hindsight is 2020. If AbbVie had known that the data was going to read out so well, they probably would have kept going. But in that time point, they had actually decided to close the trial down before then.
Mm-hmm. All right, maybe a last follow-up questions. Again, that goes with the startup creation and how you went about licensing such a candidate with a big pharma at the very beginning of a startup. So if you can comment on this, that's a question that we just received.
Yeah, I think it's a great question as well, because I think it is unusual for a company of our size and to in-license a well-characterized phase 3 program. we were actually a little skeptical that they would do it actually when we first approached them. So it is a good question. But I think we leveraged a couple of things. One, we were very, very focused on kidney disease. And to AbbVie's credit, even though they backed out of the area, they still saw that it was a huge unmet need. And there were many people at AbbVie who had worked on atrasentan for years and years, and they wanted this product to get to patients. So when we approached them with the concept of taking a drug that, you know, they knew probably had some clinical benefit, but they didn't have enough data, and repurposing that into IgA nephropathy, where we could advance it very quickly into a trial where we could get approval on a surrogate biomarker, they were extremely supportive from a patient point of view to get that drug back into the clinic because they knew they were never going to go after a rare disease in an therapy area where they didn't have a focus. So that helped a lot. We also approached them with the data. I think I mentioned that we had some data around single cell sequencing that showed some insights that were new. But then lastly, I think we had a lot of personal connections through Versant Ventures with AbbVie. I think there's a lot of trust and respect that they were putting the product into a safe set of hands. But also at Chinook, we had actually employed a number of consultants from a clinical point of view that came out of AbbVie that had worked on the program. So we showed to them that we had good capabilities to be able to advance the program forward. So I think it was a confluence of a number of factors that allowed us to license that out of AbbVie.
Very nice. Well, Tom, we have a lot more questions to ask you, and we're going to save them during the panel that you accepted to join. So that's going to be after Dr. Zinka's talk. Thank you very much for joining us.